with my age. It's come up quickly. Now, good afternoon, everyone. It is a very complex topic has been given to me, and I took a long time to study about it a bit, and still I can't understand fully. But the complexity is it is so much. I put in quite a few slides, so I will be rushing through because I want to show the data. If I can share the slides later with people, that'll be possible. Now, this is my slide deck. What is longevity, biological aging, and what is the relationship between longevity and aging in relation to the current metabolic disease, what we are facing up with? And then what are the evidence available? And I just touched base upon quite a few endocrine scenarios. So the background is Ivan and Long, 1921, as the human longevity is a very, very complex thing between genetics and environment. There are a lot of epigenetic factors comes in, and the attenuation of IGF-1 has been associated for a very long time. I'll show you some evidence of the all organisms, but it's all going to the micro level. But in humans, IGF-1 has got an age-related modulation with lower concentration in elderly. And I'll tell you how the IGF-1 decides the longevity. Now, what is aging? One has to decide what is aging. The decline in survival of frequency with advanced age caused by accumulation of macromolecules and cellular types. So when we get old, that within advance of age, there are a lot of damages happens, and it's a physiological decline, but it's a complex process, as I mentioned, with the epigenetic alteration. But I think even it is an enormous complexity, but the molecular basis looks very, very limited, and for a few highly evolutionary conservative biological mechanisms. But again, as I said, it is a complex thing. During the last 3 decades, the most discussed topic for aging and everything will be IGF-1. Accumulating evidence suggests that this pathway plays a very, very important role. You can see in the picture there that the tissue hemostasis, neural protection, insulin-like effects, and also the side effect, if you like, the IGF-1 has got is a malignancy thing as well. So where was the concept started? I think if you look at the initial evidence of growth hormone signaling in the control of human aging, it's largely indirect, and Samaras reported a huge number of examples, but it's still like a negative correlation of longevity. So when the growth hormone or IGF-1 levels are in the higher range, then the longevity is lesser. However, even it is being seen in the taller people as well, the longevity. So it is a quite complex one. But if you look at the animal models, it is shown the downregulation of growth hormone and IGF-1. So hope you understand. So downregulating the growth hormone and IGF-1 will make the lifespan to be much more. But the human data is slightly contradictory. But one can't do a human data in a big way. But I'll show you some animal studies, what is being done. So reduction of somatotropic axis has got a beneficial effect on the longevity. You can see in the diagram, the target cells has got a really good beneficial effect. What is the key mechanism? I showed you how the origin of the thing happened, but what is the key mechanism? So you look at it, the effect of reduced growth hormone signaling on aging and longevity. Why is there is inflammation reduced, the sensitivity goes up, stress resistance goes up, and then when the growth hormone signal comes down, the longevity goes up in both sides. You can see the insulin secretion, insulin sensitivity, IGF-1 levels all play a role. So the roles of IGF, if you like, I've shown you in the first, third slide or something like that, where there is a fetal development, growth in adolescent, childhood, and homeostasis is very important. We know what are the things that the growth hormone, even adult growth hormone deficiency can cause in a human being. A lot of symptoms, we replace them with after the ACDA score. But I think here, in addition of what we know, there are other effects like atherosclerotic, protective effects, neural effects, and also skeletal wise, we know the osteoporosis and everything can happen. So we need to top it up to make sure they are not growth hormone deficient. So IGF-1 system longevity in animal models. So there is a complex relationship with all this. You can see the metabolic wise, what the topic is given, almost everything is involved here. white adipose tissue. Dr. Sridhar was talking about quite a fantastic lecture about that. And then the pancreatic beta cell, thyroid glands, I'll touch base on that very, very quickly. And also about the cytokines, which is important after the COVID, and also the heart muscles and the pituitary gland, which is very, very important in managing all this. So as I said, this is the evidence that I got in vertebrate and invertebrate models. So this is a lifespan. A feedback loop may allow the cells to make some developmental or lifespan decisions. So if the environmental thing happens and then the insulin-like signaling responds to the environmental signal. So whatever happens to environment, we are definitely, our body will be sensing, and then there will be a lot of alterations will happen. Now, this is about the Drosophila Melanoglaster. We know that the common fly that we get it, 2-week lifespan of this thing. You can increase the lifespan when you suppress the receptor for insulin and IGF-1 6 times more than what normally used to live. I'll show you another thing as well later. But invertebrate models also, there is evidence to show that stimulation of IGF-1 doesn't decrease the lifespan, but when you downscale it, there will be a huge benefit. So, as I mentioned about the Drosophila Melanoglaster, if you do have the flies with the mutation of genes, it definitely causes, with all these mechanisms, the aging, the longevity, everything improves in this group of vertebrates. Now, vertebrate models, Snell's and Amy's mice, I won't go into the details because it's so more purely scientific. With PIT and PROP1, we normally do for the pituitary cases. But Snell and Amis mice are the 2 mouse strains with mutation in PIT and PROP genes respectively. And then the Amis dwarf were exposed to calorie restriction. And this is the beautiful thing about it. So when you have a calorie restriction, there is a longevity, the lifespan increases a lot. So that will almost lead to IGF-1 signaling and then the growth hormone deficiency, and therefore the longevity increases. And this is a simplified description of IGF-1 transduction pathway in the invertebrate and mammals. It's a quite complex one. I won't go into the detail, but it is happening in invertebrates and mammalian things. So the mechanisms are size, insulin sensitivity, and adiposity, but the probable mechanism is tumor resistant and cellular senescence. So here we have to understand that there is also pleiotropic benefits or effects of IGF-1 on the health status. As I mentioned in the 3rd or 4th slide, there is a mechanism of tissue homeostasis, neural protection, and skeletal homeostasis as well. The IGF-1 increases the activity of AKT protein. This is one of the important mechanisms, how IGF works and then gets the thing. So I have so far explained that IGF-1 helps in a lot when the negative feedback is there and the IGF-1 levels are less and the longevity is prolonged. But how it works, the activity of AKT protein with relevant cell survival and proliferation in glucose metabolism and protein synthesis, this is the way that it works, so that the metabolism is downscaled and the longevity is increased even though aging is progressing. So nevertheless, it's a main risk factor, as I mentioned during my talk. It can increase main risk factor of several tumors, but AKT protein activation is the one that helps with the longevity. So again, just to show how the calorie restriction will lead to IGF-1 level getting low, therefore the health span and the longevity goes up. And the decreased inflammation, FOXO is another molecule that plays huge importance in endocrine world. Now I'll show you some quick data in that on FOXO, which showed enhanced autophagy, so longevity increases. So these are the ways that the endocrine biochemical profile observed after the calorie restriction. So in the centenarians compared to younger subjects, so the centenarians has produced a good response when you have a calorie restriction. But this is the thing I mentioned about the other studies. One is IGF-1 receptor correlate with male longevity in Italian population. This is the human Trevisco longevus Trelong study. confirmed that genetic and biochemical way that Italian population has got the genetic contribution to longevity from one of the genes, IGF-1 gene. So that is the one human trial that I can show. And as I mentioned about the FOXO3, and then the low IGF-1 showed in other studies as well. So inverse relationship, when the IGF-1 levels are low, the longevity is more. And how about the second hormone, which is the thyroid hormone? The higher the TSH concentration and the lower the T4 levels, it is associated with survival benefit, no question about it. And the participants with low levels of TSH has got definitely a higher mortality rate compared to high TSH level. So the author speculated that the lower the thyroid function may lead to lower metabolic rate, and therefore the aging process comes down. But don't ask me whether you're hypothyroid being good for you, but that can't be explained. As I said, when you go up in the age, there will be a subclinical hypothyroid situation happens. It will lead to real adverse outcomes, and there is a thing that says that subclinical hypothyroidism can lead to cardiovascular problems, and it has been associated with worse outcomes in that age group actually. So how about obesity? Obesity conveys definitely a high risk of type 2 diabetes, but the age-related disease and chronic metabolic disease are all definitely relating to the obesity actually. So Dr. Sridhar has went through all this, the mechanism of aging. There will be decline in progenitor cells and cellular senescence and inflammation in obese people very quickly compared to the people who are not obese. The adipose tissue has got a real pathophysiological process relating to adipose tissue dysfunction. So once the adipose tissue dysfunction happens, that will lead to quite a lot of things, including the senile inflammation that will lead to aging very fast. So obesity, as I mentioned, that will link directly, and then the aging will be more in obese people, and the epigenetic factors will be contributed to obesity already, and that will always contribute to the longevity of a person. And type 2 diabetic, no brainer. The cross-sectional shows there are a lot of inflammatory pathway and everything, and that will lead to almost negative impact in longevity. So I'll just skip all this, that the interaction between metabolic disease and aging, there is some data to show that on molecular basis of chronic metabolic disease. So just conclude within my 14 minutes or so, the mechanism of longevity seems to be very complex. IGF-1 has got a clear link, and lower level of IGF-1, there is huge benefit. Same thing with the thyroid levels as well. So the people with high TSH levels have got less chance of getting longevity earlier when compared to the people with high TSH. So it can, when you have, I showed you evidence of vertebrate and non-vertebrates to show that a lower level of IGF-1 will lead to longevity. And also we need more data in this, but I think it is whether optimal level of IGF-1 is not known. Otherwise, we can all go into the level and live longer than what we normally do. So future is should be evaluated functional status of IGF-1. We should do that, I think, and there will be a Google and AI will come into play, will help us to go into that area and just compare. But I think there are other factors will kill us before living a longer life. Anyway, thank you very much for listening, and hope I've done justice for the time and the topic what has been given. Thank you.